The overall goal of this procedure is to permeabilize adherent cells. This is accomplished by first culturing experimental cells on cover slips in 6-well plates. The second step is to rinse the cells with 1x PBS and cover them with the solution containing the biomolecule to be transferred into the cell. Next, the inert gas jet is run over the sample, permeabilizing the cells. The final step is to fix and stain the cells if desired. Ultimately, immunofluorescence microscopy is used to show permeabilization efficiency and cell viability.
The main advantage of this technique over existing methods such as sonoporation or gene guns is that this technique is easily set up and allows for the spatially restricted permeabilization of adherent cells in a culture. Generally, individuals new to this technique may struggle because it could take time to perfect certain parameters such as the gas flow rate or capillary diameter to allow for efficient permeabilization in their particular applications.
The mass flow controller used in this procedure requires an external 12-volt power supply and is attached to a helium gas line to ensure a precise gas flow rate. Interfacing between the mass flow controller and the computer is performed by a data acquisition device. This unit is controlled by an input voltage ranging between 0 and 5 volts. A control loop in the LabVIEW program determines the required voltage at any given time. 2 positioning linear slides are used to control the movement of the 6-well plate, one for each direction. Each assembly consists of an MA15 series